Bigger cell, more membrane. So it takes things in better, right? Backwards. And this is the one mistake the whole topic is built around. The big cell is the one that starves. Growing is a losing trade, for a reason you can do in one line. Give me 3 minutes. Everything a cell needs, oxygen, glucose, ions, crosses its surface. And everything it has to keep alive sits inside, in its volume. So model it as a cube. 6 faces, each one S by S, an interior of S cubed. Divide them, and the squared and the cubed don't cancel evenly. One S survives, underneath. Start as small as we can label it. Side of 1. Now double it. Nothing changes but the size. The membrane grows in 2 directions, 4 times as much. The interior grows in 3, 8 times as much. 4 against 8. The volume wins that race every single time, and the ratio halves. Plot it at every size, and it stops being an anecdote. There's no side length where growing turns it back up. So here's the sentence that costs the marks. And the first half is true. The big cell has 4 times the membrane. On totals, it wins easily. But that isn't the question. Membrane isn't there to exist. It's there to serve an interior. And that interior went up 8-fold. So per unit of the stuff it has to feed, half as much membrane. More surface. Less surface per unit of volume. Both true, and the mistake is stopping after the first. A falling ratio does its damage in one place, the middle. Molecules cross, then random walk inward. That walk only reaches about a micrometer in. And that reach doesn't grow when the cell does. Same shell depth every time. Growth only feeds the core. By a side of 12 micrometers, more than half the cell is out of reach. And the trip scales with the square of the distance. Double the cell, quadruple the weight. So a cell running out of ratio has 2 honest moves. The first one it makes every generation. Stop growing and split. The interior didn't change. Same 64 cubic micrometers, just parceled. But 2 small boxes have more skin. Shorter side, bigger 6/S, which reframes division. It isn't only about copying DNA, but splitting isn't always on the menu. A gut lining cell's whole job needs it big. So it makes the other move. It stops being a smooth block. Each finger hands you 4 sidewalls. Its cap and its footprint cancel exactly. 100 fingers take surface from 96 to 256. Volume only creeps to 72, which is why folded membrane is everywhere. Cristae, thylakoids, root hairs, air sacs, the branches of a neuron. Everything you just watched is downstream of one line. And it isn't a cube thing. A sphere gives 3/r, a cylinder 4/r. The constant changes. The falling doesn't. Why cells stay small? Why they divide? Why they flatten? Why cristae exist? 1 ratio, 4 answers. So when a question compares 2 cells, don't answer the totals. Say, per unit of what? Then answer. Want to step through this one yourself? Links below.